Thalassemias form a genetically heterogeneous group of disorders unified by deficient production of either alpha-globin or beta-globin chains. Critically, heme synthesis itself is normal. The problem is purely in the quantity of globin chains produced. These conditions are globally widespread, concentrated heavily in Africa, India, Southeast Asia, and the Mediterranean basin. Beta thalassemia major, also known as Mediterranean anemia or Cooley anemia, results from homozygous or compound heterozygous defects in the beta-globin gene. It produces marked anemia through 4 overlapping mechanisms: reduced hemoglobin synthesis from less beta chain output, accumulation of excess insoluble alpha chains that aggregate inside red cells and destroy them prematurely, ineffective erythropoiesis, where developing cells die within the marrow before even entering the blood, and a relative deficiency of folate, driven by the hyperactive but inefficient marrow. The resulting severe anemia triggers extreme erythroid marrow expansion, physically distorting the skull, facial bones, and long bones. The red cell morphology is dramatic: marked microcytosis, hypochromia, target cells, and poikilocytes. Hemoglobin F, alpha-2, gamma-2, is elevated throughout the patient's life, which protects newborns from symptoms for approximately the first 6 months. Generalized hemosiderosis accumulates from chronic hemolysis, ineffective erythropoiesis, and repeated transfusions. Beta thalassemia minor, from heterozygous inheritance, is clinically mild, producing minimal hypochromic microcytic anemia. Its distinguishing laboratory marker is a raised hemoglobin A2, alpha-2, delta-2, which helps separate it from iron deficiency anemia and anemia of chronic disease, both of which can appear morphologically similar. Sickle cell thalassemia, from co-inheritance of hemoglobin S and a beta thalassemic allele, resembles sickle cell anemia, but is typically less severe. Alpha thalassemias are most prevalent in Southeast Asia and result from deletions of one or more of the 4 alpha-globin genes. Normally, chromosome 16 carries 2 alpha-globin gene copies on each of its 2 copies, totaling 4. Deletion of increasing numbers produces progressively worsening disease. Severe alpha thalassemia produces elevated hemoglobin BARTs, gamma-4, or hemoglobin H, beta-4. Unlike beta thalassemia major, severe alpha thalassemia is symptomatic from birth because hemoglobin F cannot be made without functioning alpha chains. Finally, hemolytic anemias caused by mechanical destruction of red cells arise when circulating erythrocytes are physically shattered by prosthetic heart valves, by fibrin strands in DIC, or by platelet-rich thrombi in thrombotic thrombocytopenic purpura. When the cause is partial occlusion of small vessels, the condition is specifically termed microangiopathic hemolytic anemia. The blood smear in these cases shows characteristic red cell fragments called schistocytes or helmet cells.